Hello, this is the group comprising of Nicholas Braca, Hannah Markham, and Eddie Barragon. This presentation will be about the composition and function of nucleic acids in a cell, specifically to do with RNA. We're going to cover and go over the different types of RNA, their function, their composition, of course, and also how they interact biochemically with um, different biochemical reactions and the effect that it has on different processes in the body, and the overall physiological value that they have to maintain basically the health of a functioning organism. So, yeah, thank you. This is Eddie Barragon representing the group, and I hope you enjoy our presentation.
The first topic we will be discussing in this presentation is the components of RNA. This is going to lay the foundation for students understanding this subject. RNA is known to be in most viruses and living organisms. It is a class of nucleic acids which we'll be further explaining later in this presentation as to why it falls into that specific category. RNA is a single-stranded molecule, which is why diseases such as COVID-19 are so deadly, because it can replicate and mutate at a much higher rate than other double-stranded molecules. The nitrogenous bases that make up the structure of RNA are adenine, cytosine, uracil, and guanine. The overall structure of the RNA molecule contains nucleotides. In further detail, it is ribose sugars attached to the phosphate backbone, with the previous bases that were just stated attached as well. As previously discussed in the last slide, this shows more detail. This picture that's included on this slide shows more details of the 4 RNA bases that are part of making the RNA strand. They are bonded to the phosphate backbone through a phosphodiester bond, and when it comes to which pyridamines and purines bond to each other, as you can see under the bonding of bases tab, this is just a shortcut way of helping students to be able to remember which goes to which. But as you can see, A goes to U, which is adenine to uracil, and G goes to C, which is guanine to cytosine.
For our check for understanding for the overall structure of RNA, what you'll do is you will write down 2 of your own test-like questions based on the 2 previous slides, so the components and the structure of RNA. Once you have written down those 2 test-like questions, you will pair up with your shoulder partner or somebody next to you. You will ask them your questions. They will then respond to you with the answer based off of the question you provide them, and then the roles will switch. Your partner will share the information to you, and then you will then answer their question.
In this slide, we will be discussing RNA synthesis. This is the overall process for RNA is transcription. A DNA template is used to make RNA by an RNA polymerase. There are 3 main steps in creating RNA. The first step is step 1 is initiation. And what happens in this step is an RNA polymerase scans the DNA template until the promoter sequence. Then step 2 is elongation. This is where the ribonucleotides are added to the template for mRNA. And the third and final step is termination, which is when the RNA polymerase hits the terminator sequence and transcription immediately stops. The picture that's included on this slide, it's just to show a little bit of the overall process that happens in RNA synthesis. But further in this presentation that we're doing today, they will be going in a little bit more detail of mRNA, tRNA, and all of that stuff further on in the presentation.
Around the room, there are 10 different DNA template strands for you to use and see. Each of you need to go around and create 4 different mRNA strands based off of the DNA template strand that is provided for you. Once you have your 4 RNA strands, you will answer the following questions that I'll be leaving up on the board with your group. The 2 questions that you will need to answer once you have your 4 mRNA strands are what enzymes were you while you were reading the DNA strand, and when the enzyme above is reading a DNA strand, what causes the attachment and detachment from the DNA strand. Within RNA, there are 3 main types of RNA. There's mRNA, tRNA, and rRNA. So the first type of RNA that we're going to be talking about is mRNA. mRNA is a single long strand of RNA that you can see over here on the right, and it is transcribed using DNA. This is going to be the RNA synthesis activity that we did, where you went from A to U, T to A, C to G, and G to C. The initial transcription is also known as the pre-mRNA, and the mRNA will take some time to mature and fully develop before it is released out into the cell. This maturing process is due to the removal of introns or non-coded sections and exons that are joined together, or the part that is being combined. There is a 5' cap, also known as 7-methylguanosine, gets added to the end. So this little dot over here on our mRNA is going to be our 5' cap, and this also holds all the genetic information required to make proteins. Our next RNA is tRNA, also known as transfer RNA. What this guy does is it will translate the mRNA into proteins, and it contains 3 different arms. You can see its general structure over here on the right. Looks like a T, so that's where it got its name. It has the D arm, the T arm, and the anticodon arm. These carry the amino acids on the 3' acceptor site into the ribosome, so it'll be this bottom site here. And the aminoacetyl tRNA synthase is a specialized enzyme that gets used to load the correct amino acid onto the specific tRNA. This amino acid is bound to the tRNA based off of the anticodon, so a set of 3 different nucleotide base pairs will be the opposite of what is located on the mRNA. And our last form of RNA is rRNA, or ribosomal RNA. That's just because it is what makes up the ribosome. And you'll see over here on our image onto the right that there are 2 different subunits. The ribosomes will have 3 different sites within this subunit for the tRNA. The site names are the first letter of what is actually occurring at each of the sites. So we have our A site, or our acceptor site. This is where the tRNA will enter into the ribosome. Then we'll have our peptidyl site, or our P site, which is where the amino acid is attached to the tRNA, will get linked to the polypeptide chain of our amino acids. And we have our exit, or our E site, which is where the tRNA will actually leave the ribosome.
So, the impact that RNA has on protein synthesis. The primary function of RNA is to facilitate the creation of proteins via the translation process of DNA replication. With all three types of RNA, being messenger RNA, rRNA, or tRNA being necessary for the synthesis and function of proteins. Ribosomes, in addition, are also responsible for the creation of various proteins through the process of translation when RNA is transcribing information through that process and replicating it into the molecule of DNA. So these are the processes involved in, different cellular activities. But essentially, without RNA and without the additional use of ribosomes, there would be no ability for the body to create proteins, which is the main takeaway. So, in conclusion, RNA is essential for the function of life as a whole. It is both a contrasting and complementary structure to the features of DNA, not only in the way it's structured in terms of DNA being a double-stranded, double helix structure and RNA being a single-stranded spiked structure, but just the fact that RNA is necessary for DNA to receive and have transcribed information, even though there's only one contrasting nucleotide, being that RNA uses uracil while DNA uses thymine. That small little biochemical difference creates a world of impact on organisms, basically allowing for higher level organisms like bacteria and multicellular organisms to have the genetic material necessary for more complex structures. And with future research, RNA shows promise to be unlocked for a variety of additional uses and functions in a whole host of fields. Just gene editing and splicing, and the use of more specific antibiotics or antiviral medications based on the antiviral loads. Yeah, there's a whole host of potential there, and that's basically what I wanted to leave off on the presentation, is the future of RNA and how much more it still has available to teach us. So I hope you enjoyed our presentation. This has been Eddie Barragon on behalf of the group. Thank you.